Welcome back to the Chapter 16 recorded lecture. I hope you didn't miss me too much since the Chapter 15 recorded lecture. Now let's begin. This diagram shows us the determination and differentiation of an embryonic precursor cell. In this case, the precursor cell is destined to become a myoblast and then into its final role as a muscle fiber. That transformation starts with a precursor cell that has the ability to become any number of cells, like a fat cell or a cartilage cell. But certain things in its environment turn them into muscle cells. That one thing is called a master gene, called MyoD. MyoD is a master regulatory gene that binds to various genes and stimulates their expression, or in other words, turns them on. Once this happens, the cell is no longer a precursor, and it is called a myoblast, which is a determined skeletal muscle cell. Once a cell has made this transformation, there is no going back, because the MyoD regulatory gene stimulates the expression of the MyoD gene, meaning that it will stay in its new state forever. This next diagram starts with a sperm and an unfertilized egg. As you can see, there are two different substances in the egg. Those substances are called cytoplasmic determinants. The cytoplasmic determinants are spread throughout the unfertilized egg unevenly. Once the cell is fertilized and divisions start to occur, the concentration of each of the determinants in the cells will be different. These varying concentrations start the differentiation process by manipulating the gene expression of even the earliest cells. In the second part, right over here, we see a cell later on in development, when it is around 32 cells in size. It is showing us the effect that cells have on each other. The cells on the bottom are sending out signals to the cells around them, changing the expression of the genes in those cells. This process is called induction. The signals received through induction typically put the cell on a developmental pathway, changing its expression and eventually its role in the overall organism. The final diagram of this lecture goes along with an experiment on Drosophila, or fruit flies. The scientists who created the experiment theorized that the bicoid protein in the fruit flies coded for the anterior end of the fruit fly. To test this, they studied mature Drosophila eggs to see where the bicoid was in the eggs. The results proved their hypothesis to be true, that the bicoid protein does in fact code for the anterior end of the fly. They tested and proved that if you were to inject bicoid into any part of the egg, anterior features, like a head, would grow from that part of the egg, or anywhere you injected it.